Now what we see pictured on this slide is what's oftentimes called the dogma of molecular biology. And what that means is that this is how all the information flows within a cell. All of the information that a cell has is housed in the DNA molecules, which are present primarily in the nucleus, and then we do have a little bit inside some other organelles like the mitochondria and the chloroplast. That DNA is something that we get from our parents, and our DNA is pretty much set when we do inherit that DNA from our parents. So that means that it's not going to change over our lifetime, with the exception of mutations and errors and things that may take place. The cell is going to take the information in the DNA, and they will choose to utilize different portions of that DNA. And when they use the DNA, they will make a copy of the DNA in the form of an RNA molecule. That process of copying the DNA into an RNA molecule is called transcription. And we will talk a bit about transcription in this chapter and the different enzymes that are involved in that. And then that RNA molecule is going to serve as the template for making the proteins inside the cell. That process of taking the RNA and translating it or turning it into a protein molecule is called translation, and we'll also talk about that process. So if we begin by talking about how we're able to take such a simple molecule, really as DNA is, and code for all of the information inside the cell. There was a lot of experimental research that went into figuring out how a DNA molecule is capable of coding for the information inside the cell. Because when you think about it, the DNA molecule itself is really quite simple. It is a polynucleotide, so it is made up of nucleotides, but it only has 4 different bases, and that's the A, T, C, and G. And somehow inside the cell is able to take those 4 letters and essentially turn them into 20 different amino acids that are going to construct the proteins. So if we think about how many bases, the A, T, C, and G, it takes to code for an amino acid, first off, it's got to be more than 1. Because if you think about this as similar to our English alphabet, keep in mind that we've got 26 letters in the alphabet, but we can certainly make a lot of words from those 26 letters. And the reason that we can is because we're taking those letters and we're reading them in more than 1 letter at a time. So we're looking at them in combinations of 2 letters at a time, or 3 letters, up to 10 letters, or even more at a time. And because we're doing that, we're able to turn them into a lot of different words, or they're able to mean a lot of different things to us. So if we go back to our 4 bases, if we were to read these one at a time, that means that you've got words essentially that are made up of 1 letter. So A, T, C, or G. That means that reading them one at a time, we get 4 different words or 4 different possibilities, which is really not enough to code for the 20 amino acids in the cell. So really what we're saying here is that we need to be able to come up with at least, at a minimum, 20 different words. So we know we can't read them one at a time. If we read them 2 at a time, basically add another blank to our word here, that means that now we can add another 4 possibilities in that second slot, and that would give us 4 times 4, or 16 possibilities, if we were to read these as 2-letter words. Again, 16 is not 20, so that's not enough. If we were to add a 3rd position, that would give us another 4. And so now we have 4 times 4 times 4, and that right there gives us 64 different words or possibilities. Now these words or possibilities would be any combination of these 4 bases, read 3 at a time. So there are a lot of different possibilities there. If you were to go through and list every single one of them, and I've just listed a few here for examples purposes, you would have 64 different ones. 64 is certainly more than 20. So the way that all of this is going to work inside the cell is that the genetic information, the information in the DNA, is encoded as a sequence of non-overlapping base triplets, or what we typically call codons. So a codon is going to be 3 nucleotides. Okay, so we're going to read 3 nucleotides at a time, and that is going to be equal to 1 codon. Now, if we look at the math in this and make sure that we understand the math. If we look at this question, suppose that hypothetically humans had 6 different bases that composed their DNA. We have A, T, C, and G, and now we're throwing in an X and a Y as well. We want to know what would be the minimum codon size required to specify or code for the 20 amino acids found in human proteins. We're going to approach this question the same way we looked at it on the previous slide. We need to come up with at least 20 codons or 20 words. So we're going to take our bases that we have, and if we read them one at a time, notice here we've got an A, T, C, G, and we've also added an X and a Y. So if we read them one at a time, we've got 6 different possibilities. 6 different possibilities is not 20, so we can't read them one at a time. It's not going to work for us. If we add yet another position, we've got again our A, T, C, G, X, or Y in this position, so that gives us another 6. And if you look at 6 times 6, we're going to get 36 possibilities, and 36 obviously is more than 20. So the answer to this question would be that we want to read them 2 at a time, or we have a codon size of 2 nucleotides. This is a hypothetical question, of course, because humans don't have 6 different bases in their DNA. They have 4. They have the A, T, C, and G. But we do want to make sure we do understand where the math is coming from in all of this. Now, if we look at how this is going to work with that flow of information, or that central dogma of molecular biology, we've got our double-stranded DNA that is going to be present inside the nucleus. Now keep in mind that DNA, it is double-stranded and it is extremely long as well. We've got all of our base pairs there. The A's and the T's are across from each other. The C's and the G's are across from each other. With the process of transcription, we're going to be looking at that DNA molecule and we're going to be making an RNA copy from it. Keep in mind that RNA is very similar to DNA in that we've got nucleotides that are composing it. We do happen to have a different sugar there. We've got ribose instead of deoxyribose. And we do have one base that's different. We've got the uracil instead of the thymine. Other than that, though, the base pairing rules are going to work exactly the same. So if you look here, we're going to be actually reading off of this top strand. And if you look at what would be across from an A, in this case now we're going to put a U because we don't have any T's in RNA. Across from the C would be a G, and so on. Across from a C would be a G, and we can look at that the whole way down. So what we're doing with the process of transcription is we're making an RNA molecule that is a copy of one of the strands of DNA. Then we're going to be looking at the RNA molecule specifically, and we're going to be reading those nucleotides as groups of 3. That's what the codon is. Those words, those groups of 3, mean specific things to the cell. What they mean to the cell is what amino acid do you need to incorporate into the protein that you're making. So if the cell were to see UGG, for example, they would know that that means a specific amino acid, tryptophan in this case. If the cell sees UUU, they know that that means a different amino acid, which happens to be phenylalanine. So when we say that the information in the DNA is present as non-overlapping groups of 3, that's what we're talking about right here, that we've got the codons. It's every group of 3 is going to mean something specific to the cell, and that's going to tell it really which amino acid needs to be incorporated into the protein that's being made. Now what we see on this slide is what is called the genetic code. The genetic code is a table that tells us exactly what each one of those 64 words means to the cell. So if we are reading our nucleotides in groups of 3, remember that we have 4 times 4 times 4, which gives us 64 different possibilities. So on here, we do have 64 codons. And each one of those codons is very specific in what it means to the cell. Now, 61 of those codons are going to tell the cell a specific amino acid that needs to be incorporated into the protein that's being made. So for example, if the cell happens to see UUU, then it knows specifically phenylalanine is the amino acid that is going to be incorporated into the protein. if the cell sees AAA, then it knows specifically that that means lysine. It's a different amino acid, needs to be incorporated into the protein. So there are 61 of those that tell the cell specifically an amino acid to incorporate into the protein. Now, one of these codons, specifically this one here, AUG, is what we call a start codon. Now, we call it a start codon because that will always be the very first codon that the cell is going to be reading through in that process of translation. Not only is it a start codon, but it does code for an amino acid. This particular amino acid happens to be the MET, or what's called methionine, that we see right here. So that means that methionine will always be the very first amino acid that we have in a protein that's being made. And that amino acid may get incorporated later on as well. So it is very possible to have a methionine at the beginning of a protein and also scattered throughout the center. Now, the 3 codons that do not code for amino acids happen to code for stop codons. So those are the ones that we see shaded in pink on this table. What that means is that if the cell happens to read one of those particular codons, that means that it can stop making protein and that it can basically go and start working on something else at this point. So notice that all of these codons that we have in this table, they all have U's inside of them, or many of them have U's, and we don't see any T's in this case. That is because the codons are going to be specifically read from the RNA copy that we make. So in that case, you're not going to see T's on the table. Now the codons, when we look at them in the RNA, they do have to be read in the correct reading frame. So if we look at how this works, if you look at this English sentence here, the red dog ate the cat, when we read that sentence, it does hopefully make some sense to us. Notice also that these are 3-letter words, which is exactly what we're looking for when we're talking about codons. Now, if we simply shift the spacing of these words, so shift it over by 1 or shift it over by 2, notice that it is changing the meaning altogether, or basically making it mean nothing to us when we read this sentence. You have exactly the same thing in the DNA. We can keep the letters in the same order, but if we don't get the groupings of 3 correct, it's not going to mean the same thing to the cell. So if we look at this piece of RNA down here, if we look at the very first line of it that we've got, we see initially we have an AUG. If we wanted to figure out what the polypeptide or protein is that we're going to make from this, we would take the AUG and we would go and find it on the table. So here we see it. AUG is our start codon, and notice that it codes for the amino acid methionine. Next, we would look up CUG. CUG is right here, and it codes for leucine. So next would be LEU. Then we look up CCA. So CCA is right here. That's going to be proline. If we look up one more, CAU, CAU is going to be over here, and it codes for histidine. Now you could go the complete way through this and read through in those groups of 3 and figure out what the polypeptide is going to be that gets made in this case. Now if we look at the next line down where the spacing has been shifted over just a little bit, if we start looking up our very first group of 3, we get a UGC. If we look up UGC, UGC is up here and it codes for cysteine. So notice that right away we're getting different amino acids than what we had before. CAC is going to be histidine. So notice it's not the same. You could do the same thing for the next line. What this is supposed to emphasize to you is that if the groupings of 3 are not correct, we're not going to get the desired protein inside the cell. So that means that the cell must spend some extra energy to make sure that it does get the groupings of 3 correct. And notice that as soon as we get the first group of 3 correct, the rest of them are going to fall into place after that. So a lot of effort has to be put into that very first selection. Now it's important to understand that we don't necessarily, or we absolutely don't start with the first 3 nucleotides that are going to be present in an RNA molecule. Now it's certainly simplified for you on these slides, but keep in mind that the RNA molecule itself may be several thousand nucleotides long, and maybe we're just going to read 1,000 nucleotides in the middle of it. What that means is that there's going to be a lot of extra nucleotides on this end, and there's going to be extra nucleotides on this end as well. So we're really just looking at the middle, and we're not picking the very first 3 nucleotides that we see. The cell will have to skim through the ones in the beginning until it defines and decides on the first group of 3 that's going to be utilized. Notice also that this last codon, the UAA, which would be the stop codon in this case, is not really going to be the last 3 nucleotides in the RNA molecule as well. It will continue on after that. Now we do want to be able to translate or take a piece of RNA and turn it into the polypeptide or protein that would be made from it. So here on this slide we have an example of an RNA sequence. Notice that our RNA molecules are single-stranded, and notice as well that they've got one end that has a 5' end, which keep in mind is the phosphate end, and they've got one end that has the 3' or the hydroxyl end to it. We want to always, no matter what, read our RNA in the 5' to 3' direction. So just in the orientation that is written on this slide. When we do that, we're going to take the very first codon, which would be AUG. We're going to look that up, and here we have it down here. It's going to start with the MET. Then we're going to look up the next one, CAU, which is over here, and that codes for histidine. We look up the next one, UCC, and UCC is going to be right here. So that's SER. And then UAA is going to be a stop codon. Okay, so there's the polypeptide that would be made. Not only are we going to read the RNA in the 5' to 3' direction, but keep in mind that there are specific ends on our polypeptide that is made as well. So the first end that is made would be what's called the amino end. And keep in mind that the amino end can also be called the N-terminus. So that lines up with the 5' end. And then on the 3' end, we will have the carboxyl end, or the carboxy, or the C-terminus. So we're going to read our RNA in the 5' to 3' direction, and we're going to make our protein in the amino to carboxy orientation. On this slide, we want to emphasize that the genetic code, that table that we've just been looking at, is nearly universal. What that means is that not only is that the table for humans to use, or plants to use, or pigs, or a tobacco plant like we see on here, but it's also what insects use, bacteria use, so it's nearly universal. Everybody goes back and uses that table when they're trying to translate or interpret the codons inside the DNA. This may seem quite simple, but this has greatly simplified the process of genetic engineering. What that means is that it is possible to take a DNA sequence out of one organism, maybe a bacterial cell, and take that piece of DNA, simply stick it or insert it into a plant or into a pig or some other organism, and they know exactly what that DNA sequence means because they read it the exact same way as the bacterial cell did. So in other words, they're all speaking the exact same language. On this slide, it's just emphasizing some of that. In the first picture, we've got a tobacco plant that's expressing a firefly gene. That means that the DNA sequence in a firefly that tells that firefly how to make that glowing protein, that DNA sequence has been taken out, it's been stuck into a tobacco plant, and now the tobacco plant knows how to make that glowing protein, and it basically does. And now it glows just like the firefly. The second picture is showing a similar thing with a piece of DNA that has been taken out of a jellyfish. A jellyfish that lives way down deep in the ocean, where it's very dark all the time, produces a protein that fluoresces. This DNA sequence for making that has been taken and inserted into the pig, and now notice that the pig has some areas that are actually fluorescing or glowing as well. So we want to make sure that we keep in mind that if a plant cell sees AAA as a codon, it is going to mean the exact same thing to that plant cell that AAA means to a bird or to a person, etc. Make sure that you understand you do not by any means need to memorize the genetic code table, but we definitely want to be able to look at it and understand how to use it to take an RNA sequence and turn that sequence into a polypeptide.